Well, we'll continue with the next presentation. And this is the last presentation of this morning. Then we're going to take a lunch break. And this afternoon, when we convene again, we'll be in the laboratory. You'll get a chance to do some hands-on with the instrumentation and try out some of these things we're talking about. This talk is, we're going to focus on eye mobility spectrometry. From now on, we'll call it IMS. And we're going to focus on the detection of the volatiles. Okay. Even though when we go back to the lab, we're going to be doing some sampling of particles and detecting the actual explosives or the drugs in particle form. We want to focus on volatiles. We want to focus on the odors and target those compounds. So in order to do that, we got to do two things: sample the odor. And you heard the canine handler. If the odor is not available, the dog can't detect it. It's got to be there. Okay, so some mass, some amount. Even though we're talking about, Patty was talking about picograms, 10 to the minus 12 grams, and nanograms, 10 to the minus 9 grams, a billionth and a trillionth of a gram. That's extremely low. The dogs can detect it, and so can our instruments. So we got to sample it first. We got to collect it. So sampling is collecting. And then we would like to preconcentrate it. So that means get as much onto a substrate as possible, so then we can take that substrate and put it on to the instrument or in the instrument. And what we've done is we've taken basically a glass fiber disc made out of glass fibers. I'm going to show you a picture of what it looks like under high magnification, and coated it with a special coating, polydimethylsiloxane. Okay, and that allows us to both sample and pre-concentrate the volatiles, those odor signature compounds that we're interested to detect. Then, once we've done that, we've got to deliver that sample to the instrument so that that detector can detect it. Now, along the way, we've had to optimize a lot of the parameters, and that was the NIJ-funded research that we proposed to them at the time, was to optimize the conditions of the instrumentation of the IMS instrument in order to detect these new target compounds, because they're not normally configured to be detected by IMS. And then we, do the things that normal analytical chemists do when they develop a new method. What are the limits of detection? How low can you detect something when you've optimized the parameters? What are sources of false positives? And what is the level of false negative alerts? Is there anything that we can't detect? And then what are the potential interferences? And then we'd like to compare the state-of-the-art. Patty referred to canines as the gold standard to this other technique, this other method. And we'd like to say that we're not going to compete with them. We're going to complement the use of canines with this new technology. Now the geometry, we call it planar. The reason we changed the geometry, or we want the geometry is important, is because it has to be compatible with the instrument. We have to get the sample into the instrument in some way. And then I'm going to talk about how does it perform. That's when the rubber hits the road. How does it work? So we're going to cover that in this presentation. And I'm going to start with volatiles. What are volatiles? When do we say something is volatile? Well, that is when the vapor pressure is high enough. That means the vapor pressure of a compound is measured in torr as standard temperature and pressure at STP. It has to be greater than 0.1 torr in order to be volatile. Okay? The equivalent of that is something that has a boiling point less than 100 degrees C. So if you have something that has a boiling point less than 100 degrees C, it's considered to be volatile. We can detect volatile compounds with our method very easily. We can also detect semi-volatile compounds. So they have a range between 0.1 and 10 to the minus 7 torr. TNT, for example, has a vapor pressure of 10 to the minus 6 torr. It is semi-volatile. It's right in the middle between something that's non-volatile, like popcorn kernel, not volatile at all. If you put a popcorn kernel on the table, it's not going to evaporate anytime soon. And something that's very volatile, like methyl benzoate. Non-volatile are things like a leaf of marijuana. That's non-volatile. THC, the active ingredient in marijuana, is not volatile. However, if you look at some of the chemical markers that we look at to associate to the presence of marijuana, that's exactly what that dog was doing. That dog was associating the presence of these compounds right here to the presence of marijuana, plant, or maybe even the presence of the active ingredient, tetrahydrocannabinol. We're doing the same thing with cocaine, not volatile, okay? But methyl benzoate is very volatile. MDMA, not volatile, we'll call it low. But Piperonal is very volatile. So we're going to target these chemical markers and associate them with the presence of, in this case, it's drugs. And we know methyl benzoate is what the dogs are alerting to because we've done a lot of studies, both in our group and other people have done studies. Here's the compound. What happens is we have cocaine. Here's the chemical structure for cocaine. We have decomposition that happens at this bond breaks, and when this bond breaks, and the combination of this methanol and this benzoic acid, there's this benzoic acid, here's methanol, they combine to form methyl benzoate. When this breaks, these bonds break, they also make this other compound called eganidine methyl ester, eganidine methyl ester, EDME. Now this also has some volatility, so it's semi-volatile. So if we target this compound right here and this compound right here, we're going to be associating it with the presence of cocaine, even though cocaine is not available. Yes, sir?
You're going to target both of them at the same time or individually?
We can. We can. The presence of this compound is always there. This is very volatile. This is not as volatile as methyl benzoate, but it's there to a lesser extent. So if we can, we will put in a program into the instrument to target both of these and say it's an alert. But we might even say, you know, if you see methyl benzoate, alert to it, and that will be the first screening process. Now, methyl benzoate is present in lots of things. So the presence of both of these together is you get more of a confident indication. Adam?
So if I'm the user and I'm looking at my screen, it's giving me a feedback, and my alert says cocaine may be present. Is this developed into an algorithm where if both items are present, then the algorithm will say with a greater degree of confidence this is? So like in my readout, I'm going to see some kind of alarm. Is it going to tell me 95% sure it's this, 98% sure, or is it just going to say there's a probable presence of this item here?
None of these screening tools give you a % confidence right now. It'll you know, there's a level above which you'll get an alert, and you can set that level to minimize the false positives, so you can make it lower or higher. That's where you get the confidence, you know, the % confidence.
All right, so I guess what I'm saying with that is, and I'm not going to look at a percentage, but let's say you say 95% is enough to trigger a presumptive identification of a material. I, as the end user, when I'm looking and I'm pushing the button on the unit, it's not going to come out and tell me I see both of these items here. It's just going to tell me that this is probably present. Whether or not it finds both items and increases its confidence, it's going to be transparent to the end user.
Yeah, so these things have not been incorporated into the alert trigger yet. Right now, you know, these instruments are designed to detect the parent compound. So you'll see a red alert, and then you'll see cocaine, right? If you've got the cocaine present. What we're working on now is saying, okay, we can associate the presence of methyl benzoate and EDME to the presence of cocaine. So we'll have to work that into the algorithm, but that's, fairly simple to do.
No, and it is. I guess what I'm getting at is there may be some environments where we may encounter an investigation where we want to know more about this material, and there may be methyl benzoate present because it's an industrial setting or something like that, and we want to have more confidence in what we're seeing to kind of pinpoint where you want to direct your investigation to shorten it up. And I was just curious if the end user will be able to see the selectivity of the instrument.
Yeah, you'll actually get to see what the alert is. So you'll get the alert for methyl benzoate, and I'll show you what the output looks like in just a couple more slides. So I anticipated your question, Adam. Patty?
Can I also comment? We also take blanks beforehand. So if you're going into a setting where you have methyl benzoate in the air, you can determine what type of level it's at. And then if you get a higher threshold or higher intensity, then you can see, okay, this area is more suspect. Because you always take a blank beforehand.
Kind of like a clean air calibration or a leveling calibration. Okay.
Yeah, the main point is that this compound right here does not have a high enough vapor pressure to be available to be detected, to be smelled by canine or by any instrument if you're doing vapor detection. You got to actually lift it off of a surface to get it into the instrument. And we've done a lot of studies over the years and published this in peer-reviewed literature where if you were to sample cocaine, pure cocaine, and collect the odors with a special instrument, this is called GC gas chromatography, and we're going to couple this to a mass spectrometer. When you sample the air above cocaine, pure cocaine, what you see is methyl benzoate, EDME, and a little bit of benzoic acid. There's no cocaine peak here. It's just not available, okay? So we're going to target these compounds. We're going to target the presence of this compound. Now this is a laboratory grade, instrument that takes a long time, 20 minutes, to go through just the analysis cycle. But we do this just to show that methyl benzoate is the target, EDME is the target. They're both there. And what we're going to do is first we got to sample it, we got to collect it, and we're going to pre-concentrate it. And in 1990, a professor at University of Waterloo in Canada came up with a really neat new tool called solid phase microextraction. And it's been used in literally, thousands of different applications for sampling and pre-concentrating. So the advantages of SPME are that we have the analyte, our target, is pre-concentrated onto the surface. It's selective. We can change the chemistry of the solid phase microextraction. It's very fast. It's very sensitive, very easy to use, and it's equilibrium based. So we have our compounds here. They evaporate if they have a high enough vapor pressure into the headspace, and then we're going to collect it. We're going to sample it with this fiber right here. And the reason it's made into a fiber is that we want to inject it into a gas chromatograph. So normally this is the geometry that's used when we want to introduce a sample into a gas chromatography instrument. There's this needle here that pierces the septum to introduce liquid into a GC. And instead of injecting a liquid, we're just going to expose this fiber right here that has our analytes in it to the instrument. So now, with the aid of some heat, all of the analytes are going to be introduced into the instrument. And this is the only formula for the whole class, so promise no more formulas. But here is the mass of the amount of analytes being collected. This is an equilibrium constant. This is the volume of the fiber, which is also constant. So we can relate the amount, this is the initial concentration of our analyte, directly to the amount collected. So we have a proportion of the amount collected in our system to the initial concentration. So that means that we can do quantitative analysis if we wanted to with this system. So back in 2004, one of my students, Jeanette Pear, developed this interface between an ion mobility spectrometer and the SPME. So we had this septum here. Excuse me. We had a septum here where we would introduce our fiber, and then we had this heating area that would heat all of the analytes off the fiber, and then those would be introduced into the IMS. Now, the next problem we had to deal with was, all right, so the instrument can detect a peak, but it doesn't know what that peak is. Now, canines don't have to be told this is cocaine or this is methyl benzoate. They just, the canines are told this is an illicit substance. You can play now, and they'll alert. The instruments will come up with some peaks, but they don't know what that is, if that's an important peak or not. So we have to tell the instrument, when you see methyl benzoate, give us an alert. And so we have to optimize the conditions for the instrument to target those compounds. And in the case of drugs, we've been working with Piperonal, methyl benzoate, and these series of compounds. Sometimes all three are detected when marijuana is present. So these are the mobilities. This is a parameter that we use. It's kind of like how long does it take for these ions to get down the tube. These are the optimum temperatures. These are some flow rates. Eventually, we optimize this, and we have a single universal parameter to run everything. And this is what they look like, Adam. When you have a plasmagram, you have a gigantic peak that looks like this. This is the reactant ion peak. And then our analytes will look like this. So the blank fiber, you have nothing down here. When you sample cocaine, you'll see methyl benzoate. When you sample pure methyl benzoate, you'll see the peak coming at the same time. So you'll get both methyl benzoate, you get after 5 milliseconds of exposure, you'll get a peak corresponding to methyl benzoate, and that's what's on cocaine. If you expose that fiber inside of a can to, say, pure cocaine, you will see the accumulation of methyl benzoate over time. So this is extraction time, how long you place the fiber inside the can. After about a couple of minutes, you get enough methyl benzoate to be detected. So this is like a 5-minute extraction, you get 2.67 nanograms. And after some time, after about 15 minutes, you know, there's no point in sampling longer because you've already saturated the SPME device. So this number may seem low, 2.6 nanograms, but this is a ton for this instrument. I don't know if you could see that, but we're looking at two peaks here. Methyl benzoate, wait, there is a white, hold on, blue. Methyl benzoate is this peak right here, and EDME is this peak right here. So we will actually see two peaks in this case. And we can train the instrument to say, when you see these two peaks, you give us a better degree of certainty that you may have cocaine here. So we'll be looking at these target compounds. In the case of marijuana, we'll see 3 peaks, right, for the presence of the marijuana. And we're still talking about relatively short extraction times with this fiber. Now, this fiber is not the optimum geometry yet, but even in about a minute of sampling the air inside of a can, we can detect enough mass of these compounds to elicit an alert from the instrument. Same thing with Piperonal. Piperonal, pure Piperonal at very, very low quantities, we get a peak that comes out right there. When we sample 2 tablets of MDMA, we get a peak right there, the same thing. We know it's Piperonal that's being detected. MDMA does not have a high enough vapor pressure. We're always going to see this. So this afternoon, when you're working with these instruments, you're going to see a giant peak called the RIP, the reactant ion peak. That's always there. We're going to let you know where to look for the important alerts. Similarly, when we're looking for Piperonal, we get this extraction curve after some time. Piperonal, we get accumulation onto the fiber. So these are fiber extractions of SPME, not optimized yet. So this is, I realize, still a long time to sample. You don't have this kind of time in the field. You don't have 20 minutes to collect the odor. That's why we're going to come back to this dose response curve for the canines. We have permeation rates in the nanogram per second. And here's the kind of response we get. As we increase the amount that's available for the dogs to alert, we increase the response. You heard the canine handler. When there's none there, when we're 0, the dog is not going to alert. It's not there. We got the same thing going on with instruments. So just make that kind of connection. As we increase the amount that's available, the more we collect and the more we introduce to the instrument, the better the chance we are going to get an alert, and the higher the peak. We're talking small, 1/1000th of a gram. 1/1000th of a gram. That is very, very small. The next thing we've got to deal with is identifying potential sources of false positives and false negatives. The canine handler says the dog's never wrong. Okay? How many believe that? I don't. You know, there are things that will elicit a false positive. Okay? So we just got to identify what those things are. We've decided to look at things that can be adsorbed onto the fiber or onto our disc, and then identify those things that are going to elicit that false response. We also have to worry about breakthrough. We're going to introduce this concept of dynamic sampling. When you place the fiber or the disc inside of a can and you close the lid and you let it sit there, that's called static. It's not moving. The air's not moving. You're just sampling the air inside the can. That's static sampling. We're going to do some of that this afternoon. Dynamic sampling is what dogs do. They're bringing their nose up against an object and they're sniffing. We're going to do that too. We're going to have a flow through our device and sample the air in a dynamic way. So one of the things you have to worry about is that you lose the compounds with this thing called breakthrough. So if you sample too much air, the compounds that were there initially, they're there in small amounts, will get adsorbed onto your fiber or onto your disc. If you sample too much air and there's not any more of your volatiles, the air will push them through. So you have to worry about that. It's not an issue for the most part. We've done a lot of testing with breakthrough, and we don't see it. Okay, but it's something to think about. This is important when you are ionizing these compounds and you've got other things. You're going to ionize your compound and these other things. If these other things are in greater proportion, they're going to compete with the ionization capabilities of the instrument, and you may not ionize your 1 nanogram when you got milligrams or micrograms of other stuff. So we got to worry about ionization. And then the other interference that we worry about is a peak showing up in the location where the analyte peak, and then it confuses the instrument, and it thinks it's methyl benzoate, but it really is something else. So we got to deal with all these things, and we've done a lot of studies in my group trying to address these potential interferences. Adam.
So help me to understand, if I was the end user and I would be using your instrument, like when you use an Ahura, if you have too much autofluorescence, then you could see on the scale it's going over your ability to see the molecular signal of the ramen. Or would it be something that the user would be able to identify by eye shot as this runs?
Yeah, it's very similar to that. You will see that giant peak, this peak right here, this will be depleted if you have ionization competition. This peak will be gone. Then you know, well, I've got other things going on here, but there's so much of this other stuff that my reactant ion peak is not there anymore. So that's an indication that you've got ionization competition. So yeah, so there are ways to detect these kind of things. Now let me go to geometry. Janus Palisian invented and patented the fiber geometry for solid phase microextractions. So you have a solution, or you could have this in air. You place your fiber, this is a special chemistry, into the environment where your sample is, and all the compounds are going to be absorbed or adsorbed onto the surface here, and then you can insert that into your instrument. So this is the sampling and preconcentration step, and then the analysis step goes later. But there are other geometries. You can take particles and coat them with the special chemistry. You can take a stir and coat it with the special chemistry. You can take a tube and coat the inside of a tube with the special chemistry and flow your sample through it. Or you can just take even your a container and coat the inside of the container and then somehow analyze that. What we did was we took a disc and coated the disc because the disc is compatible with the introduction of the sample into an ion mobility spectrometer. So we take our disc, we coat it, and then we adsorbed or absorb, we collect the sample onto the disc, and then we place it into the heated desorber, which we don't have to change the instrument at all now. It directly goes into an IMS, commercially available IMS. There's already 15,000 of these instruments available deployed out in the civilian sector. In the military, there's even more. So this thermal desorption, so we heat it up, the compounds come off of here, they enter this ionization region, and then they become ions. There's a little gate here, shutter that Patty described, pushes the ions through the gate, and then the separation happens here. Okay, so this is a size to charge separation that happens, and then we have our detector that sends a signal for the presence of a peak. So here we will have 3 peaks, one peak here, one peak here, one peak here in the plasmagram, each indicating the presence of compounds at these locations. So we went from a syringe to a planar geometry. So we're going to coat our a piece of glass, for example. And Patty developed a way to coat glass using spin coating and also dip coating, so we can control the thickness of these polydimethylsiloxane coatings that can be used to absorb the Piperonal that is present in the MDMA tablets onto the substrate. So we do the sampling, preconcentration, then we heat it up in the instrument, and this is the peak that is indicative of the presence of MDMA tablets. What we see is that when we use a fiber geometry, we get a very low signal in comparison to the planar geometry. So we have a much higher signal in terms of amount of mass detected. Here we get, for 10 tablets, we get something like 400 nanograms. 1 nanogram is enough for detection. So we got a single tablet of MDMA is enough to elicit a signal of something like 50 to 100 nanograms with a 15-minute room temperature static sampling. Adam?
For the purpose of this, how did you have the tablet in a bottle sitting in an open
All
room?
of these are in a can. So all of these is static sampling. This is static sampling inside of a can, placing our coated glass plate to sample. Okay, so this is just the research stage, okay? We haven't gotten to what's going to be useful for you guys yet. Okay? So bear with me for a second. So we published all this stuff. So first things first, you got to demonstrate that that this works. You got to demonstrate what are the limits of detection, and we got the cover of drug testing and analysis. Similarly, with explosives, we decided to focus on smokeless powders because they're widely available and they also are a threat. So we can detect these volatiles, 2,4-DNT, 2,6-DNT, ethyl centralite, diphenylamine. These are present in many smokeless powders. So we're going to target those. And same thing. Here's the reactant ion peak, RIP. Here's the time where we see the reactant ion peak. And here's a diphenylamine peak for smokeless powder. This all happens in milliseconds, okay? We talk about seconds. Here's another smokeless powder example. We get an alert for diphenylamine, we get ethyl centralite, and we get the RIP peak. Now we're getting into using planar sol gel PDMS, diphenylamine for different powders or different smokeless powders. Again, this is a static extraction curve. 15-minute extraction, we get a lot of just a couple minutes if we get 27 nanograms. And here's a study where we looked at a large number of different smokeless powders. So we had our colleagues at Federal Laboratory provide us with samples from many different manufacturers, and this is just showing the presence of different targets. So diphenylamine was present, the blue one, present in all of them. Then we look for 2,4-DNT, present in some of them, methyl centralite, ethyl centralite. So now we know what to target. Diphenylamine was a good target. And diphenylamine is collected very quickly. I'm not going to show you a lot of this data, but we also did a lot of work with FlexX. So these are plastic explosives. And what we're going to do is we're going to target volatiles that are present in plastic explosives. So some of these are military explosives. And we can see that very quickly, on the order of minutes, static extractions, we can detect these compounds. So this work was done with the Transportation Security Laboratory in Atlantic City, and we published this work. We're looking at the taggants, but again, like I said, if you've got a bad guy that doesn't use tagged military explosives, we can look at these other compounds that are in the plasticizers. And then again, you could look at the combination of them. So we published this work. And here's what we're going to do. A is the bare glass fiber disc, how it looks like. This is the surface. This is the surface of the bare disc. When we get done coating it with our technology, it looks like this. So the thickness goes from being around 275 microns to being 350 microns. So we've increased it, the thickness, and it looks totally different. Basically, it's a plastic polymer coating that has an affinity for organic compounds. And if you have, and you can get a flow through this. So if you've got a flow of air going through this, and you've got your methyl benzoate or you got your diphenylamine, they will come in contact with this coating and they will stick there. Now it's our job to get this over into our instrument for detection. So this is the surface, what it looks like uncoated, and this is what it looks like coated. Here's a even higher magnification view of the coating. This is plastic. This is polydimethyl siloxane polymer, and it's almost like a liquid. And when the compounds come in contact with that liquid, the liquid will absorb it. And then you can release it when you heat it up again. So when we're doing dynamic sampling, which is what you like to do, suck some air through this coating with a pump, we're only talking about a 20-second exposure. So here's smokeless powder. We're going to target 2,4-DNT, providing relatively small amount of exposure of the volatiles, 14 nanograms per second. And using a very low volume pump, 0.17 liters per second, that's a handheld pump, we can detect 2,4-DNT in smokeless powder with a 20-second exposure. So what does this translate into the real world? Well, we've been working with some shipping companies, and they have loaned us some of these LD3 containers, and we want to sample these containers both statically, that means place our device inside the container, and then analyze it before it goes off, before the plane takes off. Okay, so we got some time for that. And dynamically. So here's our pump. Our device is over here at the end. And we're going to sample at some distance away from where a very small amount of material is. 500 milligrams of smokeless powder, half a gram of smokeless powder. We can detect this right at the mouth of the can, but also some centimeters away from the can. We can sample short period of time and get a signal even as far away as 5 feet away from the can. Okay? So here are the extraction times. We're talking about seconds. Okay? So sampling on the order of 30 seconds or so. We can detect TATP. Here's a signal. For the TATP work, we had to go to the University of Rhode Island. One of my colleagues, Jimmy Oxley, has the ability to manufacture, synthesize, and make TATP. We had a very small amount of TATP, 10 milligrams. 5-second extraction, dynamic extraction with our fiber. Here's what it looks like now. This is the commercial version of it. It has this little holder. The SPME actually goes here. It's a white looking, looks like a glass disc. Goes in here, and if you sample for 5 seconds, you can get a huge peak of TATP by sampling just 10 milligrams using dynamic extraction. So what do we talk about? IMS can be used for volatiles. Hasn't been up until now for the most part. We need to identify what are the target compounds. We can use SPME and p-SPME for sampling and preconcentration, but we have to deliver what we've sampled to the instrument. We've had to optimize the parameters of the instrument, and we have done a lot of work already to identify the false positives and the false negatives and potential interferences. Even though I haven't shown that data to you, it's been published. And we say that IMS can complement the canines. Okay, we're not going to compete. They're not going to replace the canines. Here's an additional tool in your toolkit. This novel geometry does offer improvements over the fiber geometry that's already been used in thousands of different applications, mainly environmental applications. And it is amenable and compatible with IMS. That's why we're interested in that geometry. And it also enhances the sensitivity. We can absorb a lot more quantity of material by using this planar geometry as opposed to the fiber. So I do want to acknowledge financial support from several folks. Primarily, NIJ has funded this effort, but we've also received some funding from NIST and the University of Rhode Island, as well as the NIJ Technology Center of Excellence to fund this particular workshop. A lot of people have been involved over the years. Here's some collaborators. For example, the people at the DHS S&T laboratory, people who have provided smokeless powder samples, been analyzed, people who have provided drugs for us, and people who have helped us with sampling of these explosives that normally we wouldn't be able to get our hands on, like TATP. And then, of course, all the students who actually do the work, like Patty, but there have been others. A couple of weeks ago, we had Wen Fan here, Mimi Young here, Howard Holness here, and a postdoc, Dr. Essler, who's recently joined our group. But these are the people who've been working on IMS research in my lab over the last, I would say, 8 years or so. And then again, here are the papers you can always go back to and read some of the, it's impossible to show all of the data that supports a lot of this work, but if anybody's interested, the peer-reviewed publications are there, and now there are probably about a dozen just coming from our group. And other people, other groups are now publishing using our technology. Questions?
I guess one question is whether you've looked into heating up the sample. Because there are techniques where the can itself would be heated up a little bit.
To assist the static sampling. Yeah, that's very commonly used when we're doing fire debris analysis in order to assist with the evaporation of those compounds to get a better sample. Now, the normal situation out in the field is going to be static sampling at whatever temperature the container is in. So you're not going to be able to heat an LD3 container very much. But we do know that heating the sample does help to assist with the evaporation. So we're going to be doing two things. We're going to sample two ways in the lab. Static, just so you see what it looks like, in the can. But normally static sampling will happen in a big container. So there's a big problem now is how do you sample shipping containers, the very large shipping containers? Can we place our device into a metal box on a corner of the shipping container and let it sit there over the course of a voyage? And then when it gets to port, you take it out and then you do the analysis. That's one idea. The other is dynamic sampling. So these containers now all have a hole where you can put a hose on and sample the air through the hole. So we're going to talk about both of these sampling methods, static and dynamic. You're going to be using both of those. For vehicle, we have an exercise doing vehicles. We're going to hang some of these strips inside of the trunk. We're going to sample the vehicle trunk over some time. But we're also going to, sniff the air out of the inside of the vehicle. Currently, what they do with vehicles is they'll swab the steering wheel, the shift changer, the door handles. So they're looking for particles. We're going to take it one step further. We're going to look for the volatiles inside the vehicle.